Hi, my name is Ken Lasserson. I've written a site called Microbiome Prescription, a free site with a lot of microbiome information. I've been asked to do a quick presentation, somewhat focused towards a MD or researcher, to try to explain what the model I'm using is based on, where it's going, what it can be used for. And I refer to it as the multi-dimensions of health. And the reason I'm saying multi-dimension is that often with medical health issues, things are reduced to a single dimension. And with the microbiome, that model basically does not get results. So let's go on and take a look at the first slide. First slide is what I call the one-dimensional model. Infection happens, labs are done, positive results in a lab. Infection could be replaced by a diagnosis, but for the moment, let's just keep it at infection so that it's a standard model. And with infection, you also have symptoms. The symptoms may cause an MD to skip doing labs for a variety of reasons. One is the time to get labs there and back, the person is already over it, or if it's something like cellulitis and pretty acute, he needs antibiotics right now, there's not time enough to wait for it. So there's an assumption that a certain type of infection, prescribe antibiotics for it, and then see if you match or not match. My own personal experience, I've had cellulitis and they picked the wrong antibiotics originally, and when the lab results came back, they changed to the correct one. So labs and symptoms often drives treatment without labs. Sometimes to save money, sometimes the lab's not available, sometimes the lab results are going to come in just too late, so the MD is forced to fly after the symptoms. Treatment happens. It could either be run its course like a normal cold virus, or it could be use of anti-infection agents, for example, antivirals or antibacteria. The person is recovered, and definition of recover is usually negative lab results. They may still have some symptoms, and usually the doctor will say, they're very quaint, oh, those symptoms will clean up and remove in time, or will fade away in time. It's just leftovers, mainly because they really don't understand or have not been taught in medical school why symptoms persist even when the lab results are negative. You end up with people going back to normal or not. There's three conditions which I find very illustrative, mainly because they tend to be very prone to post-infection syndrome, and they are Giardia, and I will make reference explicitly to in a region large community outbreak in Bergen, which I always view as a sweet one because it's a national health plan. So the result is the researchers are able to do a cohort examination over time, many years in fact, of people who had it and what happens to them afterwards in terms of other medical conditions that develop, etc. And just basically sweet data, which is often the problem with medicine, is it ends up being very limited in scope. A few places like Iceland, where you have a beautiful DNA base, or many Scandinavian countries where you have a unified medical system, you are able to do much better research than in a slightly ad hoc American or even Canadian system. So Giardia is one, Lyme disease is another one. Lyme disease generally can be detected, treated very quickly and effectively, and generally recovery is good, but some don't recover. And then COVID-19, and COVID-19 getting treated, but a percentage developed what's called long COVID. So that's a one-dimensional model. Let's go over and jump ahead into a long-term medical conditions. What we find if we review the literature on the U.S. National Library of Medicine, commonly called PubMed, is study after study confirmed there are distinctive microbiome shifts seen in tons and tons of medical conditions, from brain trauma, which is a bit of a surprise, celiac disease, which is no surprise, Alzheimer's, Crohn's disease, again, not surprised. So there's a whole variety of things. But what tends to be unresolved is a series of questions. Was the microbiome a contributor to the condition developed in the first place? In other words, did the microbiome predispose somebody to it? For something like diabetes type 2, pretty strong evidence from the studies that bad eating, which of course determine a particular type of microbiome, resulted in diabetes type 2. So there's some things where the overwhelming amount of evidence suggests that indeed it does. Any condition where lifestyle in terms of eating habits is associated with disease, we can basically come to a relatively safe conclusion that the microbiome was a significant contributor to the condition. Other factors to the condition could be DNA, and co-infection, other types of infections. For example, certain types of viral infections will cause or can cause cancer or higher incidence of cancer. So there's a hodgepodge of factors that could all be significant. Second one, which often tends to be assumed without very much evidence, is that the condition changed the microbiome. That is, for something like chronic, like cancer. Oh, the cancer resulted in the microbiome changes because of the cancer impact upon the body. That one, actually, I find the evidence of that happening is pretty sparse, although it is often taken as a answer because it's an easy answer, so you can ignore the microbiome. Now, evidence-based is where I'm at, and I just don't find a hard amount of evidence for very many of them. The third option is there feedback between the condition and the microbiome? Do the two of them bounce back and forth? The condition encourages certain microbiome shifts. Those shifts now encourage the condition to proliferate stronger, etc. So there's a feedback cycle, which is the third option. The question is basically a chicken and egg, and we also have to toss in a chick into it or some other thing. So it may not be either or, it may be both and other factors involved. If you want to see a longer list of the associated bacteria with associated microbiome shift, there's a link there, and the link will connect you to each one of the studies finding that there are distinctive, statistically significant microbiome shifts seen with a particular medical condition. So that gets us to the next interesting issue, which is what I call off-label coincidence. And basically it means that many drugs or supplements found to improve a condition, almost nothing improve 100% of everybody. There are responders and there are non-responders. For a bunch of them, particularly those which the family has been affected by, and friends affected by, I've gone in and looked at the drug or supplement, some cases supplements, or what has been able to attenuate symptoms. And what I found that about 40% of these drugs or supplements, we can locate a study showing the impact on the microbiome. This is where it gets interesting. About 80% found that the drug corrected some or many of the microbiome shifts reported with this medical condition. Is this coincidence or is something else going on? 15% we find that there are no matching bacteria, which is not surprising. Often the sample sets are relatively small and often aggregated, and as you see, often symptoms are critical for identifying matches much more than the official diagnosis, which incorporates dozens different symptoms into a group who given a diagnosis. And then about 5% of the case, the drug increases the shift in the wrong way. So what we have is an interesting coincidence. Was the drug success due to the microbiome impact and that alone, or was the other impact, or a combination of the two? Did it cause certain cells in the body to do it directly, or did it alter the microbiome, so the microbiome produces different chemicals that causes the cells that change to be different? Very hard to test, interesting prospect, but it is something which becomes important. The premise which I'm working on, and people working with me is that it is both, with the key aspect of the microbiome being a very significant factor. Implication of this is that we may be able to, and probably should, set up a clinical trial saying whether or not microbiome testing will predict responders or non-responders. As in, if we have a substance called it agnostia, and we know what agnostia impacts, and we have conditions called disbelief. So we go and in our sample of 100 people with disbelief, find the microbiome, take the agnostia, test it on control group, and find out how it shifts the microbiome. And then we go and give agnostia to disbelief and see how many respond or do not respond. My gut feeling, to use expression, is that what we will find is that the responders are predominantly the ones which have the bacteria that agnostia acts upon. Very simple model. Technically, it's a relatively easy one to do a clinical study on, because you have agnostia, you have the control group, who responds, what bacteria do they have, and then you have the cohort of people with the condition, take their microbiome, see who responds and who doesn't respond, put the data together, and voila, you may have an interesting study to publish. Now, let's go on to our original model. And in this case, first of all, I'm going to move myself off screen, or to a different portion of the screen. Okay, we have a normal person, we have infections in labs. The real ideal situation would be if we have a whole population or a group of people, we know the microbiome before there, we now go and infect them with a particular disease, for example, Giardia or other things, a very easy to acquire disease. And now we, every day or every couple of hours, take the microbiome samples as they start developing symptoms and see whether or not we find that there is a pattern of the microbiome shifts to the symptoms. Of course, there's a slight problem. Willfully making people sick is not generally acceptable standards. We do have something which was recently published, which was for COVID-19, and it came pretty close to this criteria, because what they were looking at is how does the gut bacteria respond with COVID-19 infections and after COVID-19 is technically missing. During the process, they actually just found some interesting things in terms of symptoms. You have respiratory positive and respiratory negative, as in these are people who have COVID infections, universal agreement, they have COVID infection, positive PCR results, but one has respiratory issues and one do not. And what we now find, what we see is very simple. Those who do have respiratory symptoms or not, you have two very contrasting microbiome populations, which is pretty convincing evidence that some symptoms may be very much connected to the microbiome. Same virus, in this case, this is a Chinese study, pretty similar DNA study, unlike America where you have a real hodgepodge of DNA there. So you have a bunch of factors nicely controlled. Here's a study, you can go and look it up, 2019, June 2021, so it is a contemporary study. Fascinating study. There are a couple more COVID-related studies dealing with the microbiome, which are out there, which are well worth the reading. Main aspect is symptom having a very strong association to the microbiome with COVID, which is very informative. Now, let's go on and get back to what I've been working on in terms of doing research for the last 3 to 4 years. And what we have found is that when you're dealing with the microbiome, toss out the diagnosis. Toss it out. It will be more a spoiler for getting results. It will be a distractor. It's basically a gigantic red herring which is rotted. So what we find is when you look at a diagnosis, you find that almost every diagnosis has a wide variety of symptoms and lab results. You won't find everybody with identical same lab results. They're heterogeneous. And often subgroups can be identified. And generally, in terms of research, the subgroups are deferred to future studies. One key reason is you need to get a cohort of this particular subgroup to do a study on. And often it's enough of a problem to get the diagnosis cohort assembled. There's more something which occurs in only 10% of that, because you got even greater problems getting a sufficient cohort size to get statistical significance coming out. So subgroups tends to often be acknowledged there, and generally it's deferred to future studies. In other words, basically ignore, that's nice, we're not going down that way, we just can't handle it, we just don't, whatever, excuses, whatever. Now, we have over 2,000 microbiome samples from people around the world and uploaded to our analysis site, which is a public open access site, including the ability for you to go and download all the microbiome results and the symptoms they have been annotated with. So it's there, it's completely open. People can replicate what we have done to the best of their statistical abilities. And what we find is that we have 373 symptoms which has significant associations to the microbiome patterns. One of which is sort of interesting. We find that we have 4 age ranges which we detected significant patterns with. 0 to 10, 10 to 20, 30 to 40, 40 to 50. And the z-score for each one of those is at least 5.94. In other words, it jumps out. Now, you may say, okay, why don't you have 20 to 30? 20 or 30 is probably pretty close to the population norms, hence it doesn't stick out. 50 and up probably doesn't stick out because once you get in the 50s and early 60s, what we end up doing is we have so many auxiliary medical conditions causing so much noise in the microbiome that you will not get a distinctive pattern for 50 and 60 years old up. This type of information is confirmed if you go into PubMed and look at age patterns for the microbiome. It's there. Wherever the patterns match, it's never questioned, but we are finding there are patterns, and the literature finds that there are patterns of the microbiome. We also, using just data and an algorithm we developed, we found that if a new microbiome comes in, we seem to have over 50% correct prediction of the top 20 symptoms just from the bacteria, knowing nothing about the patients at all. Sometimes it can be the age, sometimes even the nature of their work, how far they walk every day. All those actually shows up in the data as distinctive signature with statistical significance. Our basic hypothesis is that in terms of a clinical diagnosis, subgroups actually may be determined by the microbiome rather than any other conditions. Hence, if you approach treatment from the point of view of treating the symptoms and not the diagnosis, you're going to probably have better results. In some ways, by working from the microbiome, you effectively are doing symptom relief on steroids. You're identifying what precisely, and you can get a very targeted set of suggestions to normalize or to correct the biome shift that are likely causing the symptoms, and thereby producing better relief, rather than saying, oh, he has a headache, take Anacin or take something else, which may or may not work for everybody, and most doctors know it only works for a percentage. Here we are improving the odds by being absolutely explicit, but by some ways being symptom relief on steroids. Okay, now I want to switch over to effectively talking about post-infection syndrome. This is particularly where the microbiome is probably most easily tested and most helpful, because generally post-infection syndrome has no effective treatment. To me, one of the best studies I've ever seen on this matter has actually come out of Norway. They had a massive community-wide Giardia infection. That, plus having a nationwide medical system, makes tracking and researching so easy, because they know everybody who had it, good big population. They're all treated. You can verify if the person indeed has had the Giardia treated, and what are the subsequent medical conditions or issues they're facing though. And what we find is that there are a small group of diagnoses these recovered patients ended up acquiring, far higher than in the population as a whole. And the interesting thing is the new diagnoses all have strong associations to ones which are known to have microbiome dysfunctions. Go over to COVID-19 long haulers. They have a very similar set of symptoms to ME/CFS, and in the Bergen Giardia patient cohort, guess what? Chronic fatigue syndrome shows up in that group. A couple of studies there, and basically there's growing consensus that COVID, long COVID, and chronic fatigue syndrome ME are the same. There is some political issues here, because ME/CFS is not treatable, and you have a whole bunch of people demanding treatment for long COVID, and the government is wanting to do it, and if they don't deal with it or do it, this will be horrendous social costs for social support and welfare. So there is somewhat an attitude of, yes, it's there, a few people are saying it's there, but let's pretend it's a completely different condition, because it is politically advised not to toss long COVID in with ME/CFS, because the latter is basically untreatable, high suicide rate, etc. Let's go on and take a look at the Bergen studies. And here I have done a selection. As you can see, it goes over many years, and you can see the magic word on all of them. All of them are dealing with the Bergen presentation. Looks at chronic fatigue syndrome 5 years afterwards. In other words, long COVID could easily last 5 years. Number one is 6 years after. We have irritable bowel syndrome showing up, and we even have overactive bladder syndrome starting to show up after 6 years. So we are seeing a long-term persistence, which I interpret as long-term persistence of microbiome dysfunction. What we don't have is we don't have any microbiome studies coming out of the Bergen ones so far. Hopefully somebody will persuade them to do microbiome and see if there is commonality with those who have the symptoms. I would expect they will, simply from the fact that we find microbiome distinctive patterns for both IBS and for chronic fatigue syndrome. And IBS is is microbiome associated, no problem. Chronic fatigue syndrome being microbiome associated, sometimes it's hard because often we still have a very outdated attitude that it's a psychological condition, not a biological condition, despite the evidence such as we see here that it is actually biological, and long COVID is in the same kettle of fish. Okay, let's try doing a bit of a recap. And the recap is trying to explain the model which we've been working on, which we think the evidence supports well. And the premise is most infections attempt to infection form its environment. Think about sci-fi, right? They're talking about terraforming our planet. That is, here's a planet like Mars, not enough atmosphere, not enough water, whatever. We go in and we go to change to make it looking more like Earth. Why would we want to do it? It's in our own self-interest. It allows us to have a population grow. Why would an infection do it? Perhaps not for cognitive thinking, but from the reality that bacteria or viruses which work in this manner survive better, so hence it ends up going that direction, almost ingrained in its RNA. I won't say DNA because if you're doing a virus, it's largely RNA. So it attempts to infection form an environment. How does it do it? Generally, it does by sending or inducing chemical signals to the body. The virus may infect the cell, and now it basically reprograms the cell to start pumping out chemicals which often are needed for the virus to replicate, or which helps the virus replicate by shutting down immune response systems. Doesn't matter, they'll go both ways, because what the infection is concerned about is proliferation. So it could do it by getting more food in, or by killing off hostiles, in other words, the body immune system. So infection starts almost immediately. The body starts being infection formed. With the infection forming, things are disrupted. You get different chemicals metabolites and enzymes being pumped into the body. Suddenly you get a headache because one of them perhaps may be a vascular constrictor, and you're not getting as much oxygen to the brain. And you develop brain fog, all because a virus is directly or indirectly pumping in signals to the body that results in it. Now, after the infection, what happens? If the infection is eliminated, all the infection-induced signals stop. Okay, good, we're done. Not quite. The problem is the bacteria shifts, often mainly the bacteria which have been adapted to this new infection form environment, like it. And they prefer this new world. It may be, for example, less oxygen environment, and the bacteria happens to prefer low oxygen. So it's going to cooperate with maintaining the status quo that the infection established. The king is dead, but the country lives on. The bacteria will often send out signals for self-preservation, and often a self-replicating feedback cycle may get established. So the microbiome stays infection form to match the original infection environment, or pretty close to it, because the bacteria which has been persuaded to join the infection are happy where they are. The symptoms persist because the metabolites imbalances persists. And metabolites is a pretty broad. It includes proteins, you name it. It's a very complicated environment. Now, the interesting thing is this concept is in the literature back in the 50s, particularly the Pasteur Institute for Tropical Medicine often used the term an occult infection because the person is still sick. And it looks like a particular infection, particularly in the literature I'm referring to. It looks like the person still has a rickettsia infection, but all the lab tests comes back negative, but they still have it. So it's a occult, almost like it's a ghost in the body. If you look at the model here, you recognize, wait a minute, aren't we saying the same thing, just different terms? And we now have a model for why it could happen, whereas they did not in the 50s have such a model to explain things. Let's go back and look at a revised view. And the infection sends messages to the bacteria. The bacteria, through a variety of mechanisms such as quorum, sensing, etc., causes other bacteria to grow. It's an altered microbiome. The microbiome starts doing feedback. The net result is we get the symptoms. The symptoms shows up here. Over time, when we have the recovery happen, the bacteria is still there, and the alterations end up being slowly reversed and you get full recovery, so you have a happy patient. Or you go into post-infection syndrome where the disruption, the feedback loop, is able to self-maintain itself. And the key thing is, remember, this is very multi-dimensional. It consists of metabolites, bacterians, proteins, other chemicals. So there's a lot of things in the microbiome soup. Now, but wait, we have a problem, but wait, there is a possible path to deal with it. Is that we know that bacteria are sensitive to the environment. They need the right food. Most of the research out there is that some substance X alters bacteria A, B, C, and D, taking some up, taking some down. If we take that and we flip the research and say, okay, we have a condition where A, B, C, and D are altered. Let's look at the literature and find out what alters them back to where they should be. In which case, okay, simple logical progression. So the result is, if we know that the shift causes symptoms, we can try to compensate for the shift and hopefully reduce or eliminate the symptoms. For myself personally, it has worked very well, and I tend to be a firm believer, but not on the soapbox at Hyde Park preaching it, but it's there. Other people I've worked with also have experienced the same thing. Some cases within 1 or 2 days of the right substance based on the microbiome, dramatic improvement happened. Okay, here's where the gotcha. Different groups of bacteria can produce the same symptoms. Think about it as crime. Symptom is a crime. It could be prostitution, it could be drug running, etc. So what's the crime family? That's a problem. You can have different groups of people, different groups of bacteria. It's a crime family: Sicilian, Irish, Russian, Nigerian, etc. Each member of the crime family will cross-support other members of the group. Nice. Nothing extraordinary about that. Similarly, members of a particular group of bacteria will cross-support it. Often, the grouping of a bacteria cartel is based by tolerance of the group's bacteriocins, which are bacteria, antibiotics produced by the bacteria themselves. So if you're not affected by it, you'll probably join the group. And you may not be affected by it, but you may produce some of it, in which case, hey, the group is all happy with you. You coming in with your 6 sutures going away, you have the right connections to the right distributors, etc. So everybody is happy. But in order to address it, we need to target and identify, not that there is a crime family behind the drug family. We need to know about the drug family. Traditionally, in the US, it's being which ethnic groups. Sometimes it can be other groups or other characteristics. For example, it could be all upper-class spoiled brats have formed a criminal drug smuggling cartel. So what that means is that we need to get the individual's microbiome to correctly target. If we know which bacteria are involved, we can now be explicit to their microbiome. In other words, we cannot just treat a symptom. We actually see a symptom. So, okay, let's get a microbiome sample so we know what is likely causing the symptom. And then we will make adjustments either by drugs, by diet, by supplements, by herbs and spices to alter the, to correct the microbiome shift which we see in the microbiome, which are probably causing the symptoms. Okay, so that's basically a overview of what I am the one I'm working on behind the scenes for the Microbiome Prescription website, and some of the labs who are making use of it in terms of coming back with recommendations to MDs who send in who uses those specific labs for diagnosis. Okay,